Welcome to our training on adverse childhood experiences and toxic stress. This module will discuss research questions relevant to our work in this area. The purpose of this module is to give an overview of the scientific method and different ways to conduct a study, define epidemiology, give an overview of types of research studies, to understand the epidemiological and biological research questions related to ACEs, and to look at some diagnostic tools and biomarkers for ACEs research. According to the definition in the dictionary, science is knowledge about or the study of the natural world based on facts learned through experiments and observation. Science helps build explanations that are consistent and predictive. Science is based on knowledge of facts, developing theories, testing hypotheses, and published and repeatable procedures. The goal of scientific research is to describe, explain, predict, and potentially manage or fix a phenomenon. In our case, we want to be able to describe, explain, and predict what toxic stress is. Our goal is to be able to create interventions to help fight against it. To achieve these goals, scientists rely on the scientific method. The scientific method is a set of assumptions, attitudes, and procedures that guide researchers in creating questions to investigate, in generating evidence, and in drawing conclusions. Let's take a look at the different steps in the scientific method. Our first step would be to develop a testable question, a hypothesis. For example, we might start with a question like this. Do ACEs result in a higher incidence in diabetes in children under 12? There are things that you need at the beginning of the scientific method. 1. A hypothesis. A hypothesis is a tentative statement that describes the relationship between two or more variables. 2. A variable. Variables are factors that can vary or change in ways that can be observed, measured, and verified. 3. An operational definition. An operational definition defines the variable in terms of how the factor is to be measured, manipulated, or changed. Our hypothesis might look something like this. We believe that children who endure toxic stress, 4 or more ACEs before a child is 12 years of age, will result in a higher sugar intake, causing obesity and diabetes. Our variable is the sugar intake. The children may or may not take in more sugar if they have 4 or more ACEs. And if they do eat more sugar, they may or may not become obese. The next important step is to begin the process of designing a study. There are a couple of common ways to design a study: experimental and descriptive. Descriptive methods are research strategies for observing and describing behavior. Commonly used descriptive methods include naturalistic observation, watching and observing and recording what you see, conducting surveys, documenting case studies or specific stories, and correlational studies. A correlational study examines how strongly two variables are related to or associated with each other. The experimental method is used to show that changing one factor causes a change in a second factor. For example, the clinicians tell patients to do yoga as an intervention. Then they check with the clients later to see if yoga helps them to feel better. Step 3. Analyze the data and draw conclusions. Statistics are mathematical methods used to summarize, analyze, and draw conclusions about the data researchers collect. A statistical technique called meta-analysis involves reviewing and pooling the results of many research studies into a single analysis. Statistics help us determine if the two things we're studying are related. Scientists use mathematical procedures to determine the frequency and prevalence and ratios of diseases. Step 4. Report the findings in a peer-reviewed publication, like a medical journal. For instance, Dr. Burke published her study in the Journal of Child Abuse and Neglect. When she reported her findings, she described the precise details of the study to make it possible for other investigators to replicate or repeat the study in order to increase scientific confidence in the accuracy of her original findings. Now we have an idea of how scientists agree to do their research. Let's take a closer look at the tools they use to prove things. Studies. When you initially begin a research project, you have an idea or a theory of something you want to prove. The definition of a theory is a possible explanation for observed findings, results from a total of findings of individual studies, a tool for explaining observed behavior. For instance, with the clinical team, a child might say their stomach hurts in the morning. A mental health clinician may have a theory based in psychology. They might theorize that the child is suffering from some sort of stress. They'll need to test if the child is hungry or not in order to prove their theory. There are different ways to study to capture information to support a theory. Let's review some of those now. To conduct studies around the questions we have around ACEs, there are three main types of research studies we'll utilize at CYW: case control studies, cross-sectional studies, and longitudinal studies. The way to tell the difference between the three is to think about it from the vantage point of time. Cross-sectional studies examine a whole population or subset at one point in time. For example, how many children reported ACEs at Baby Child Health Center between 2007 and 2009? We only collect the data once. Case control studies are looking back in time. So, for example, is there a difference between the number of ACEs between obese and non-obese children? Longitudinal studies are over a long period of time. Longitudinal studies examine a group of participants over a long time with repeated observations. For example, we can measure biomarkers of stress in the same group of children every year starting in 2014. A disadvantage of this research design is the length of time it takes to collect data and analyze it. Let's take a look at some of the research methods scientists use to come to their conclusions. Descriptive research methods are approaches for observing and describing behavior. A descriptive study describes a set of facts, doesn't look for relationships between the facts, doesn't predict what may influence the facts, and may or may not include empirical data. For example, you could measure the percentage of new students from out of state each year since 1980. One descriptive research method you could use is a survey. A survey is a questionnaire or interview designed to investigate the behaviors, attitudes, and opinions of a particular group. Surveys allow researchers to gather information from a much larger group of people than is possible with other research methods. The survey we use here at CYW is the ACEs questionnaire. Doctors, caseworkers, and wellness nurses administer the surveys to patients and base their interventions based on the results. Surveys are carefully designed questionnaires. Sometimes they're in paper and pencil format. They can be on computer, internet-based, or conducted over the telephone or even in person. Qualitative interviews are open-ended questions that the interviewee can respond to. A person gets to tell their story. Usually the interviews are one-on-one. They can also be done in groups. For example, how do you think Bayview Hunters Point people will perceive research is one of the questions asked during some of our qualitative research. There's also the case study method. The case study method is a highly detailed description of a single individual, generally used to investigate rare, unusual, or extreme conditions. An example of a case study in our work would be Anir. Anir was a girl who came into foster care with 6 ACEs, 12 years old, depressed, and not interacting. Within 1 year of good nurturance and a steady home life, she was more vibrant, interacting with others, and participating in extracurricular activities. She no longer exhibited signs of depression. One way a researcher can find out information is to figure out how interventions help people. The researcher can have one group of people who receive an intervention and one group of people that don't. That's called the control group. The intervention can be many things. Our interventions will be wellness-based: yoga, biofeedback, therapy, exercise, encouraging people to have a healthy diet. After the control group is finished and the study is over and the intervention has proven to be beneficial, the control group can also use the intervention and take advantage of the new findings. People's information can be recorded. When you go to the clinic, medical professionals enter patient information into a system. Researchers can collect medical data for a group of patients and analyze the data all at once. They can look for patterns. For instance, they might look at people with only 1 ACE to see if they're smokers in adulthood. This is called secondary data. We don't have to actively collect it. Dr. Burke used over 700 medical records in her study. Things you might find in a medical record that could be useful, like an ACEs score, height, weight, medications taken, diagnoses, blood pressure, emergency room visits, and types of diseases. When conducting research, you have to keep in mind, research is scientific but not perfect. There are and can be issues. There are some inherent sources of bias. For instance, a person participating in a research project may change or alter their responses because they might want to please the researcher or get the answer right. Or there can be subtle things the researcher does, like smiling when a particular answer is given. It can be unconscious, and it can result in answers from the participant that aren't altogether accurate. Scientists do their best to keep biases out of scientific data. They make special effort to think about every step of the research process. They take into account and consideration as many factors as possible, and design the questions carefully to ensure the participants are comfortable so they can get accurate answers to their questions. Just as we mentioned before, the scientific method is a set of assumptions and ideas that guide researchers. They also have a set of ethical guidelines they follow to ensure the safety of the participants of a study. These guidelines are born out of human suffering. During World War II, Nazi doctors conducted as many as 30 different types of experiments on concentration camp inmates. They performed these studies without the consent of the victims, who suffered indescribable pain, mutilation, permanent disability, or in many cases, death. At the Nuremberg, or doctors' trial, which brought 23 German doctors to trial right after the war, prosecutors found 15 defendants guilty of war crimes and crimes against humanity. 7 of them were hung. A stateside example of why we have the need for ethical guidelines is the Tuskegee experiment. In 1932, the Public Health Service, working with the Tuskegee Institute, began a study to record the natural history of syphilis in hopes of justifying treatment programs for blacks. It was called the Tuskegee Study of Untreated Syphilis in the Negro Male. The study initially involved 600 black men, 399 with syphilis, 201 who did not have the disease. The study was conducted without the benefit of patients' informed consent. Researchers told the men that they were being treated for bad blood, a local term used to describe several ailments including syphilis, anemia, and fatigue. In truth, they didn't receive the proper treatment needed to cure their illness. In exchange for taking part in the study, the men received free medical exams, free meals, and burial insurance. Although originally projected to last 6 months, the study actually went on for 40 years. The ethical guideline written as a result of the World War II Nazi war crimes against humanity is called the Nuremberg Code. It instituted guidelines to protect study participants by requiring informed consent, information about the study procedures, require findings be published, and most importantly, the fact that the researchers should do no harm to the participants of the study. Let's review what kinds of questions we want to have answered here at CYW. Epidemiological questions. First, let's define epidemiology. Epidemiology is the branch of medicine that deals with the incidence, distribution, and possible control of diseases and other factors relating to health. Or you might say, it's the study of how disease spreads and can be controlled. An epidemiologist is a public health professional who investigates patterns and causes of diseases and injury in humans. They seek to reduce the risk and occurrence of negative health outcomes through research, community education, and health policy. Epidemiologists work in offices and laboratories, usually at health departments for state and local governments, in hospitals, and at colleges and universities. Some do field work to conduct interviews and collect samples for analysis. To research epidemiological questions, we must have accurate testing tools. So our first research project addresses whether our ACE score questionnaire is valid and reliable. Meaning, does it accurately and consistently measure ACEs in the pediatric setting? Is it measuring what we want it to measure? Are the results we get from the questionnaire consistent? Another question is whether there's an association between one type of ACE and a specific health problem. Is there a relationship between how long someone has experienced an ACE and a particular health problem? The ACEs questionnaire will be piloted with our clinic before we extend it to the general public. Some additional questions have been added: school bullying, parent or caregiver being deported, and being treated badly because of your race and/or sexual orientation. Resilience. According to Merriam-Webster, the definition of resilience is the ability to become strong, healthy, or successful again after something bad happens, or the ability of something to return to its original shape after it's been pulled, stretched, pressed, or bent. Certainly, we can apply this idea when it comes to trauma. How does someone become healthy and strong or successful again after something bad happens? This is one of the fundamental questions we're going to try to answer. Let's take a moment to understand biomarkers. This is any measurable substance in a person that indicates a disease, infection, or environmental exposure. Biomarkers are measured from collecting cells or tissues from any part of the human body: blood, plasma, serum, saliva, buccal cells, hair, urine, feces, teeth, or sweat. For instance, a biomarker for diabetes is high glucose levels or high sugar levels. Here at CYW, we'll be looking at a few types of biomarkers when we begin conducting our research. One is CRP, or C-reactive protein, which is produced by the liver. The level of CRP rises when there's inflammation throughout the body. Interleukin-6, or IL-6, is one of the pro-inflammatory cytokines that induce inflammation, and cortisol, a long-term stress hormone. These existing biomarkers need to be proven to be accurate markers for toxic stress in association with ACEs. Biomarkers can lead to the discovery of what happens in the body when it comes to ACEs, and this can lead to the development of new diagnostics and/or new treatments. We have a couple of fundamental biological questions when it comes to researching ACEs. One, can we diagnose the severity of ACEs using biomarkers? 2, can we use biomarkers to follow up and measure how effective our treatment is with children exposed to ACEs? 3, which biomarkers should we use for certain diseases? All in all, biomarkers can help researchers understand the bodily pathways of ACEs and help develop better treatments. To conduct studies measuring biomarkers in the clinic and community setting, it helps to have specimen collection techniques that are minimally invasive. Such tools are generally called point-of-care diagnostics. They are easy to use and produce results in a short time without needing to be sent to a laboratory. Examples in everyday life are glucose monitors and pregnancy tests. Another useful technique to measure the physiologic impact of stress and trauma is neuroimaging. Rather than collecting biospecimens, we collect images of the brain that show us both structure and function. We can then compare images, biomarkers, and reported experiences of stress and maltreatment to produce a more comprehensive understanding of a child's experience of stress and the impacts on the body and brain development. Developments in neuroimaging technology, near-infrared spectroscopy, have made it more portable and less expensive to use than ever before, so it's more feasible to use in the clinic setting. Let's review our module takeaways. Scientists rely on the scientific method to conduct research. There are many unanswered questions about ACEs, toxic stress, and resilience. Case control, cohort, and longitudinal studies are useful for researching ACEs. Biomarkers are measurable substances that indicate disease and can be used to measure toxic stress. Neuroimaging, such as functional MRI and near-infrared spectrometry, can help measure the impacts of stress on the brain. We have a couple of fundamental biological and epidemiological questions when it comes to researching ACEs. 1, can we diagnose the severity of ACEs using biomarkers? 2, can we use biomarkers to follow up and measure how effective our treatment is with children exposed to ACEs? And 3, which biomarkers should we use for certain diseases? Now you have an idea of the epidemiological and biological questions our research team will explore here at CYW, as well as the types of studies we'll use to answer those questions. Through identifying biomarkers, using neuroimaging and point-of-care diagnostics, we can begin to develop interventions that will help children and families with ACEs. This concludes our module on research questions. Thank you so much for participating in this training. Center for Youth Wellness, where health begins with hope.